Welcome to Learning Made Easy, a channel dedicated to simplifying complex concepts and making them easy to understand. Today, we're going to be talking about enzymes, their types, kinetics, and types of enzyme inhibition. Enzymes. Enzymes are biological molecules that catalyze chemical reactions in living organisms. They are specific to a particular substrate and catalyze a specific chemical reaction. They are made up of amino acids and are folded into a specific three-dimensional structure that allows them to interact with their substrate. Enzymes increase the rate of chemical reactions by lowering the activation energy needed for the reaction to occur. This means that enzymes make it easier for the reactant molecules to come together and react, allowing the reaction to proceed more quickly. Types of enzymes. Enzymes can be classified into 6 main types based on the chemical reactions they catalyze: oxidoreductases, transferases, hydrolyses, lyases, isomerases, and ligases. 1. Oxidoreductases. It catalyzes oxidation-reduction reactions, for example, alcohol dehydrogenase that catalyzes the conversion of ethanol to acetaldehyde. 2. Transferases. It catalyzes the transfer of a functional group, for example, aminotransferases that catalyze transfer of an amino group from one molecule to another. 3. Hydrolyses. It catalyzes the hydrolysis of a substrate, for example, enzymes like lactase, amylase, and lipase that catalyze the hydrolysis of lactose, starches, and fats respectively. 4. Lyases. It catalyzes the cleavage of a substrate without hydrolysis or oxidation, for example, decarboxylase enzymes that catalyze the removal of a carboxyl group from a substrate. 5. Isomerases. It catalyzes the isomerization of a substrate, for example, glucose 6-phosphate isomerase that catalyzes the conversion of glucose 6-phosphate to fructose 6-phosphate. 6. Ligases. It catalyzes the ligation of two molecules, for example, DNA ligase that catalyzes the joining of two DNA strands. Enzyme kinetics. Enzyme kinetics describes the rate of an enzyme-catalyzed reaction and how it is influenced by various factors such as substrate concentration, enzyme concentration, and temperature. The Michaelis-Menten equation describes the kinetics of enzymes. And it states that the rate of an enzyme-catalyzed reaction is directly proportional to the concentration of the substrate and the enzyme. The equation is V equals Vmax, S, Km plus, S, where V is the rate of the reaction. Vmax is the maximum velocity of the enzyme. S is the substrate concentration. Km is the substrate concentration at which the enzyme is operating at half of its maximum velocity. Factors that affect enzyme kinetics. 1. Substrate concentration. The rate of an enzyme-catalyzed reaction is directly proportional to the concentration of the substrate. As the substrate concentration increases, the rate of the reaction also increases. However, there is a point at which the rate of reaction reaches a maximum, called Vmax, and further increases in substrate concentration do not result in significant increase in the rate of reaction. 2. Enzyme concentration. The rate of an enzyme-catalyzed reaction is directly proportional to the concentration of the enzyme. As the enzyme concentration increases, the rate of the reaction also increases. However, there is a point at which the rate of reaction reaches a maximum, called Vmax, and further increases in enzyme concentration do not result in significant increase in the rate of reaction. 3. Temperature. The rate of an enzyme-catalyzed reaction is affected by temperature. As the temperature increases, the rate of the reaction also increases. However, there is a specific temperature range in which the enzyme works best, called the optimum temperature. Beyond this range, the rate of the reaction decreases as the enzyme denatures or loses its activity due to the effect of temperature. For phone, the rate of an enzyme-catalyzed reaction is affected by pH. Each enzyme has a specific pH range in which it works best, called the optimum pH. Beyond this range, the rate of the reaction decreases as the enzyme denatures or loses its activity due to the effect of pH. 5 cofactors and coenzymes. Some enzymes require the presence of specific cofactors or coenzymes in order to function properly. Cofactors are inorganic molecules, for example, metal ions such as Zn, Fe, and Mg that are required for enzymes to catalyze certain reactions. Coenzymes are organic molecules, for example, NAD+ and Fad that are required for enzymes to catalyze certain reactions. 6 inhibitors. Inhibitors are molecules that bind to enzymes and reduce or completely prevent enzyme activity. Types of enzyme inhibition. Enzyme inhibition is another important aspect of enzyme kinetics. Enzyme inhibition can be classified into 2 main types: competitive inhibition and non-competitive inhibition. In competitive inhibition, the inhibitor molecule binds to the active site of the enzyme, preventing the substrate from binding. This can be overcome by increasing the substrate concentration, as the substrate will outcompete the inhibitor for the active site. Competitive inhibitors can be overcome by increasing the substrate concentration. An example of competitive inhibition is the inhibition of the enzyme acetylcholinesterase by the pesticide malathion. On the other hand, non-competitive inhibition occurs when an inhibitor molecule binds to a different site on the enzyme, called an allosteric site, which causes a conformational change in the enzyme that affects its activity. Non-competitive inhibition can either increase or decrease enzyme activity, depending on the type of allosteric site and the type of inhibitor. An example of non-competitive inhibition is the inhibition of the enzyme hemoglobin by the compound carbon monoxide. In conclusion, enzymes are essential biological molecules that catalyze chemical reactions in living organisms. Thank you for watching, and we hope you learned something new.